Hello, could somebody confirm you can hear me?
Yeah,
Thank you. We got to chapter 66. Yeah, we didn't do that. I think we're actually behind schedule. Let's go to chapter 6.
we had just finished chapter 6. We
Did we
hadn't started
finish
chapter.
chapter 6?
Yeah, we ran a little bit over and finished it.
Well, that's good.
Haven't started anything on chapter 8 yet.
All right, let's shut that down. Yeah. All right, sorry for being late. I had to confirm my existence and I don't know what you call it, double log in or something like that, verify that I'm existing, that I'm John Volpe. So that took me a little while to log in. Let's go to the syllabus. You're probably not seeing my screen, are you? No.
No.
All right, you should be seeing my screen now. Let me check my volume. Looks good. Let me blow that up. So today is November 14th, and we should be already in chapter 8, but we're just going to be starting chapter 8. Let me warn you that there's a quiz this week on Wednesday. There will be quiz 4. It'll cover lab 7 and chapter 5 and 6 of the lecture. Any question about any of that?
Nope.
Okay. um, and then we're going to have a lab today. It'll be lab 9. You're also supposed to be working on your unknown project. Let's see. Anything else I need to talk about? Your infectious disease paper should have been turned in. All right, let's go on to chapter 8. And thank you for your assistance in reminding me where we were. So this is chapter 8. This slide is showing you the major goals and a rough lesson outline. Know the terms. What's a genome? A gene, a genotype, a phenotype. What is vertical and horizontal gene transfer? What is an operon? What is a promoter? What is an operator? What is a structural gene? What is crossing over? What is transformation, conjugation, and transduction? To be able to describe DNA replication, RNA transcription, and protein translation. We'll talk about all of those in this lesson. And then 3, understand the different types of mutations. Any question about what we're going to be covering? All right, we're going to start talking about the structure and function of the genetic material. We'll talk about the flow of genetic information. We'll then move on into talking about DNA replication. Following that will be RNA transcription and then protein translation. After that, probably not today, but maybe in the next class, we'll talk about the regulation of bacterial gene expression. We'll talk about how that happens with an operon. We'll then move into mutations and talk about how you can have genetic transfer and recombination. And then lastly, we'll end talking about genes and evolution. Any question about what we're covering? All right. Genetics is the study of what genes are, how they carry information, and how that information is expressed, and how genes are replicated. A gene is a segment of DNA that encodes a functional product. That product is usually a protein, but it can also be RNA. Like all of the tRNA genes, all they do is they code a RNA, a tRNA. A gene is a region of DNA that codes for something, and there's a chromosome, and you can see the DNA wrapped up in the chromosome, and then there's gene 1, and after that is gene 2. You note that there is some region, at least on eukaryote chromosomes, that does not code for, well, doesn't code for a product like a protein or an RNA. And that would be this region here. And there's a chromosome. Let me blow that up so you can see it a little better. There's a chromosome, and we have mapped different locations of different genes. And there are they all there. You notice this region here doesn't have a whole lot mapped to it. Come on. Ah, gosh. Sorry, my computer's froze up here. How do I end this? It's going to force me to close that down. Why is this up here? Checking to make sure I don't have something running that I don't want running. I guess I should keep that up. All right, let's try that again. All right, the genome is all the genetic material in a cell. We're talking about microbes, where a cell is essentially the microorganism. So for microbes, the genome is also all the genetic material in an organism. The study of genomes is called genomics. A genotype is the genes of an organism. For example, this individual has blue eyes, and that's the phenotype, the expression of the genes. The individual has the genotype, lowercase b, meaning humans have two alleles or two genes for each physical characteristic if it's a simple genetic trait. And you don't need to know about that part. But the genotype for that individual will be lowercase b. The phenotype is the blue eyes, the expression of the genes. For brown-eyed individuals, of course, the phenotype is they have brown eyes. And their genotype would be either capital B, lowercase b, or capital B. Any question about any of that? So the genotype codes for the phenotype, at least of a simple genetic trait, and we're not going to talk about any genetic traits which is not simple. Like a person's weight is not a simple genetic trait. Okay, any question about any of that? All right, there's the chromosome of E. coli, and it's really kind of like a string. And then there would be a gene like maybe right here. DNA is a polymer of nucleotides. DNA has 4 nucleotides: adenine, thymine, cytosine, and guanine. And you can either learn it by those names, or you can call it A for adenine, T for thymine, C for cytosine, and G for guanine. DNA is a double helix associated with proteins to make the chromosome, meaning a chromosome, which is true for both eukaryotes and prokaryotes, is made up of both DNA and protein. Any question about any of that? Now, for eukaryotes, the main protein in the chromosome is the histone protein. For prokaryotes, they lack the histone protein. So they don't have any histones with their DNA. A histone is a protein, by the way. So DNA is a double helix, and one single strand of DNA is held together by chemically covalent chemical bonds, and it's held together by the sugar shown here, and then the phosphate backbone shown there. Let me blow that up. So one single strand of DNA is shown here, and it's held together by the sugar phosphate backbone. The nucleotides are on the steps of the DNA. They do not hold the single strand together. The nucleotides hydrogen bond with their pair, their covalent pair, meaning A will pair with T and T with A, and C will pair with G and G with C. And it's these hydrogen bonds between the nucleotide bases that hold the two strands of DNA together. So the two strands are held together only by hydrogen bonds between their complementary nucleotide bases. If you remember, a hydrogen bond is very weak and a chemically covalent bond is very strong. So if we were to heat the DNA up, you would only have single strand DNA. And I'm talking about heating it to 70 degrees. Jesus, that must be Celsius, 70 degrees Celsius. And I don't know what temperature that is Fahrenheit, something like 160 degrees Fahrenheit. I don't know. You'd have to look it up. What happens when you heat it is you break these hydrogen bonds. These hydrogen bonds are weak and they break and reform all the time. So, for example, this hydrogen bond here can break, but then the DNA will be held together by all the other hydrogen bonds in the DNA, because there's a lot of them in DNA. And then this will reform, and then that one will break. At least at our body temperature, 37 degrees Celsius, 98.6 degrees Celsius, these hydrogen bonds in our double strand of DNA are breaking and reforming all the time. And we call that the DNA breathes, because one bond will break, but the DNA is held together by the other bonds. And then that one will form, reform, and then this one will break. The two strands of DNA are not running together. They are anti-parallel. That means one of them is facing this way and one of them is facing that way. Specifically, you could say this is the 3' end and that's the 5' end. However, on this strand, this is the 3' end and that's the 5' end. The numbers is what the chemists do when they're talking about sugars or some chemical molecule. And this carbon here, let's see, it'd be this one here, is the 3' carbon, And this carbon here is the 5' carbon. Why they say this is the 3' end, and then this is the 5' end, because this carbon here is the 5th carbon in the sugar here. Any question about any of that? You do not need to know the 3' and the 5' end of DNA, but you do need to know that the two strands of DNA are anti-parallel. Any question about any of that? All right. In the central dogma of biology, we say that the information in DNA flows to RNA in transcription, and it moves to the protein in translation. So the information encoded in the DNA and the genes of the DNA can be changed into the information in RNA by transcription. And then that information is encoded in the protein after translation. Each of them, the DNA, the RNA, and the proteins, have their own language, which they store the information in. And we'll talk a little bit more about that. Any questions about the central dogma? Now, the central dogma used to be called the law in biology. However, they found that in certain viruses, the information in RNA can actually flow backwards to DNA. And we will talk about that when we talk about viruses. But generally, the information always flows from DNA, at least in a normal cell, one that's not infected by a virus, DNA to RNA to protein. It never flows the other way in a normal cell. All right, within a cell, we can have the information of DNA flow from DNA to RNA to protein. This we call the flow of information within a cell. It is also called the is that the horizontal gene transfer? Let me think about that. Or is that the vertical gene transfer? Sorry, I'm drawing a blank here. I think this is the horizontal gene transfer, but it's not terribly important. The idea is the flow of information within a cell. You can also have the flow of information between generations of cells. And for humans, we'd say that's the flow of generation from parents to offspring. So with a cell, you have a parent cell, that DNA can be replicated, and then the DNA will move to opposite ends of the cell, and then the cell will split down the middle, with each daughter cell getting a copy of the information from the parent cell. And so the flow of information between generations of cells, or between generations in people, happen from the parent or the parent cell to the daughter cells by DNA replication. And then the cell divides. Any question about that? And if I remember this correctly, which I might not, this is the vertical flow of information. You can also have the flow of information from a donor cell flowing to a recipient cell. And this we call the flow of information between cells of the same generation. Usually what happens is the DNA leaves this donor cell. Usually that happens when the cell dies. And then the DNA is naked DNA, and it can be taken up by a recipient cell, which we call the recombinant cell. And the light purple is the DNA taken up by this cell. And then that DNA can become a part of the chromosome of the recombinant cell. And so this cell gains DNA from another cell, which it gained from that cell. And you'll notice I broke this graph because I think my way, and I added that in, explains it a little clearer. And that is the DNA leaves this cell, the donor cell, and then it's taken up by the recombinant cell. Any question about any of that? All right, we talked about the central dogma before, and I mentioned that the information was stored in languages in the DNA, the RNA, and the protein. The language of DNA is the 4 nucleotides of DNA. And so we can say there are 4 letters in the alphabet of DNA. In RNA, the same information is stored in the nucleotides of RNA. And once again, there are 4 different nucleotides in RNA. So there are 4 letters in the alphabet of RNA. Now, when the information moves to the protein, the information is stored in the language of amino acids. And there are 20 different letters in the protein, meaning there's 20 different amino acids. For comparison with English, we have 26 letters in our alphabet. Any question about any of that? So in DNA, you could say the information is stored in the DNA as that is a dog. And in RNA, that information is stored in like a different dialect of that is a dog, the DNA, but it is still the same language. And you'd say something like, that is a dog, in the same language, but a slightly different dialect. In the RNA, going to the protein, let me back up, shrink that down a little bit. The information would be stored, and it's the same information, but in a totally different language. It would be something like quello e un cane. Same information, but in a different language. Does anyone recognize this language? I once had a student tell me it might be Italian, and they said it was not Spanish. And I was saying it looks something like Spanish, but quello I'm not familiar with. So it's probably a Romance language. Nobody's commenting, so we'll move on. All right, the flow of information can happen within a cell or between generations of cell. When we're talking about between generations of cells, I got it right. That is the vertical flow of information, where the information moves from a parent cell to the daughter cells. Or in humans, we could say that flow of information moves from the DNA of the parents to the DNA of the offspring. And that happens through DNA replication. And then in cells, that'll happen when the cell divides. But with humans, that would happen in, in fertilization of the egg and the sperm. All right, so let's talk about DNA replication. DNA replication is semi-conservative. That means that when we have our original 2 strands of DNA, they separate into 2 single strands of DNA, and then 1 strand is used as a template to make the newly formed strand. So the DNA polymerase will be reading this strand here and it'll see a T. And so it'll put the A on the newly synthesized strand using the T as the template to put in the A. And then the DNA polymerase will move down and we'll see a G and we'll add a C. And then it'll move down and see a C and add a G. So one strand is used as the template to make the newly made strand. And then this strand will be used as a template to make this strand. Semi-conservative DNA replication. Any question about that? So this is showing you the black strands are the original parent strand of DNA in semi-conservative replication. One strand will be used as a template to make the new strand, which is shown in white here. And then the offspring cell will get one original strand, which came from the parent, and then one newly replicated strand. Semi-conservative replication. This differs from the conservative model of DNA replication, where people propose that one cell would have two strands of newly replicated DNA, and then the other cell would have two strands of original parent cell, or two black strands of DNA. The conservative model of DNA replication does not occur. It's the semi-conservative model of DNA replication that actually occurs. And don't ask me what the liberal model of DNA replication is, because I don't know. How this happens is when the strand, the parent strand, is being used as a template to make the new strand. A nucleotide triphosphate, meaning in this case, a T with 3 Ps, comes in, and then the cell, the helicase that's adding it, removes the 2 phosphates here, and that releases a lot of energy. If you remember, this bond here and that bond there are high energy phosphate bonds, and if you break it, it releases a lot of energy. When the 2 phosphates are removed, that releases a lot of energy, and then that energy is used to finalize the reaction, which will actually link this phosphate to this sugar to form the covalent bond between the phosphate and the sugar. And then the T will hydrogen bond with the A. Any question about any of that? And so there it's being added, and the 2 phosphates are being broken off, and then the T is added with the phosphate sugar being the backbone of the DNA. Any question about any of that? All right, let's watch a little video on DNA replication. I'll probably shut all those down.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions about any of that? Let me keep this up just in
Is
case
done by
we
an enzyme
talk
called
about this.
3' end. Strand
There we go.
is done by an enzyme.
So it's actually a helicase that opens up the two strands of DNA to make a single strand of DNA. So that enzyme is important. And then the DNA polymerase is important. Maybe that's it there. That's putting in the primase. So let's put it right here, about right here. There it is. The DNA polymerase is the enzyme which actually adds the nucleotides to the new strand of DNA. So the new strand of DNA will go here, and the polymerase will put in whatever, I guess red's supposed to bind to green. So we'll put in the red nucleotide here. And then the third important enzyme that is you need to know is the helicase. You don't need to know about primase. Let me see where it's got shown. There it is. DNA ligase seals the fragment of DNA, meaning if there's a break here and then there's a strand here, DNA ligase will connect this gap in the DNA, meaning between this nucleotide and that nucleotide, for example. I'll talk a little bit more about that. Let's go back to here. We're going to talk about DNA replication in greater detail now. DNA is copied by DNA polymerase. The DNA is made in the 5' to 3' direction. DNA replication is initiated by an RNA primer. On one strand, the DNA polymerase just moves down the strand because the strand is in the correct orientation to make this strand, the new strand, in the 5' to 3' direction. And that we call the leading strand. In which case, wherever the DNA binds, it'll just move down that strand and then synthesize the new strand of DNA. Let's say this DNA polymerase falls off right here. And so this new strand is right to here. And what will happen is another DNA polymerase will bind right here, and it will start the synthesis of the leading strand once again. And it'll just move here, going down the strand, the helicase opening up the DNA to make it single-stranded. And then the DNA polymerase will just move down the strand. And as long as it doesn't fall off, it will make the newly replicated strand from here in one batch. So there will be no breaks in the DNA. So the only break is where this DNA polymerase bound, and of course, where initially the DNA polymerase fell off. And so on the leading strand, the helicase doesn't have to do much work. All it has to do is bind right here and then link this strand of DNA to this newly replicated strand of DNA. Any question about any of that? Now, the lagging strand is running in the wrong orientation. And so when the DNA polymerase binds to this strand, it cannot move this way because that's not the right way. It has to move this way. And so what it does is it makes the DNA in small batches called Okazaki fragments, after the doctor who discovered this. And this DNA polymerase bound here and then ran to there, making an Okazaki fragment right here. And obviously it doesn't go any further because this is the newly replicated strand of DNA right there. And then there will be another DNA polymerase that'll bind here, and it'll make an Okazaki fragment up to here. And then this one will bind here, and it'll make the DNA to here. And so the lagging strand is made in many small batches that we call Okazaki fragments. This DNA molecule here will need to be hooked together with the ligase once right here, once right here, once right here, once right here. So essentially every Okazaki fragment will have to be ligated together with the fragment behind it and the fragment in front of it. So the ligase will have to work quite a bit on the lagging DNA strand. Any question about any of that? And what the ligase is doing is it's making the single strand of DNA one single strand, which has no breaks in it, meaning it chemically puts a covalent bond in this break here. Any question about any of that? All right, so I mentioned DNA is made in the 5' to 3' direction. It's initiated by the RNA primer. The leading strand is synthesized continuously. The lagging strand is synthesized discontinuously in several chunks that we call the Okazaki fragments. Now, the RNA primers, which start the synthesis of DNA, have to be removed, and the Okazaki fragments have to be joined together. and the any breaks in the DNA have to be joined together, and that will happen by the DNA ligase. The RNA primers are actually removed by the DNA polymerase. All right, any questions about DNA replication? Remember, there are three essential enzymes: helicase, DNA polymerase, and then ligase. All right, if there's no questions about DNA replication, let's move on to RNA transcription. RNA transcription is the synthesis of RNA from DNA. DNA is transcribed to make RNA, and there are three main types of RNA: messenger RNA, tRNA, or rRNA. So when the RNA polymerase is transcribing the DNA, how does it know to make messenger RNA or tRNA or rRNA? How does the RNA polymerase know which RNA to make? Anyone have a guess? Anyone know anything about genetics at all? All right. So if I ask you a question, if the RNA is being transcribed for a gene, which RNA it makes, all you have to do is say the RNA polymerase doesn't need to know which RNA to make, because all it's doing is copying the information in the DNA. And the DNA will code for either messenger RNA, tRNA, or rRNA. If the gene codes for a protein, and most genes do, then the DNA will be transcribed as messenger RNA. However, if the gene codes for tRNA, then the DNA will be transcribed as tRNA. And if the gene codes for rRNA, then the gene will be transcribed as rRNA. Any question about any of that? Well, here's another question for you. What's the difference between messenger RNA, tRNA, and rRNA? Are they different nucleotides? Are there different chemistry, makeup of messenger RNA, tRNA, and rRNA? Meaning, does messenger RNA have a phosphate and tRNA does not? Something like that. Anyone have a guess? There's no difference between the RNA. Did I say that right? There's no difference between the RNA of messenger RNA, tRNA, and rRNA other than the sequence of the nucleotides. If the gene is being coded as messenger RNA, it's only the sequence of those RNA that determine that's a messenger RNA. And if the gene is being coded as tRNA, it's only the sequence of the RNA nucleotides that make it a tRNA. There is no difference between the RNA, a messenger RNA, tRNA, and rRNA, other than the sequence of the nucleotides. Any question about any of that? That's kind of important in genetics, but to you guys is not that critical. Transcription of DNA begins at the promoter. And I should mention that not all of the DNA is transcribed. What part of the DNA is transcribed? Anyone? I'm not going to say to this one, it should be obvious because I've already stated it. So you guys should think about that because you'll probably get a question on that, either the quiz or the final. And that is, what part of DNA is transcribed? Transcription begins when RNA polymerase binds to the promoter. A promoter, let me see if I've got a map of DNA someplace. Well, that's replication. I will mention that all of the DNA is replicated, but only some of the DNA is transcribed. When you have a DNA molecule, RNA polymerase will bind to the DNA at the promoter. So this region of the DNA, assuming this is the start of RNA transcription, that this would be the promoter where the RNA polymerase binds to the DNA. And that's all the promoter codes for. It's a region of DNA that codes for the RNA polymerase to bind to it. So the promoter is a region of DNA, and all it's doing is coding for RNA polymerase to bind to this region. The RNA polymerase will then move in the 5' to 3' direction. So RNA will be made in the 5' to 3' direction. And then the RNA polymerase will continue transcribing the RNA from the DNA until the RNA polymerase reaches the terminator. And the terminator codes for the RNA polymerase to fall off. So transcription stops when the RNA polymerase reaches the terminator. So before a gene, we have a region of DNA we call the promoter, and that's where the RNA polymerase binds to the DNA. And then the RNA polymerase moves down the DNA, transcribing the gene until the RNA polymerase reaches the terminator. And obviously the terminator is at the end of the gene, and the terminator codes for the RNA polymerase to fall off the DNA. And that will stop transcription. All right, this is actually showing you it's actually a fluorescent microscopic image, electron microscopic image of DNA, RNA polymerase, and right there you can see RNA starting to be made. So we're looking at the start of transcription. Any question about any of that? Let me see if I can get this link to work. You think? Yeah, there it goes. I knew there was something you had to do to get that to work.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial.
We'll watch that later. That's not the video I wanted. see. Although I think, well, I can't do both, so. I'm looking for a short one, but I'm not seeing. There's a time. That one might work. Let's try this one. After the commercial.
As owner of Mint Mobile, I always want to find ways to save you money. So let's meet the barely paid spokesperson.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. The motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule.
This doesn't look like it's moving the movie. Let me try that again.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. The motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly-A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons.
Okay, we're not going to talk about that now. We'll talk about that later. All right, any question about any of that? All right, so let's go through RNA transcription. Transcription begins when the RNA polymerase binds to the promoter, and then the RNA polymerase moves down the DNA. Obviously, it makes it single-strand DNA. Helicase makes it single-strand DNA, and then the RNA polymerase moves down the DNA. And that transcribes the RNA. Let me blow this up. You can see that initially the green is the RNA. Initially, the RNA is pairing to the DNA, but later on, the RNA is not pairing to the DNA. And that's the single strand, other strand of DNA. And this is the single strand of DNA that's being transcribed. And the RNA polymerase moves down the DNA until it reaches the terminator, which would be at the end of the gene. Once the RNA polymerase reaches the terminator, the RNA polymerase falls off the DNA. That will release the RNA that has been transcribed, and then the DNA will snap back and become double-stranded again. Any question about RNA transcription? If there's no questions about RNA transcription, let's talk about protein translation. Protein translation is the synthesis of proteins by ribosomes using the information in the messenger RNA. Now, if the gene codes for a protein, it will be transcribed as messenger RNA. And then that messenger RNA will move to a ribosome, or the ribosome will move towards it, doesn't really matter. And then the ribosome will bind to the RNA and then move down the RNA, sort of reading the messenger RNA to perform protein translation. Now, I said the ribosome moves down the RNA. The ribosome does not really read the messenger RNA. What happens is tRNA comes in and reads the messenger RNA. And there are 3 RNAs which are important in protein translation. There is the messenger RNA, of course, taking the information of the gene to make the protein. So that's in the messenger RNA. And then there's ribosomal RNA, or rRNA, and that is in the ribosome. Now, ribosomes are both protein and rRNA. And the protein of a ribosome is actually called ribosomal protein, or R protein. And then the RNA is rRNA. That's the second RNA. The third RNA that's important in protein translation is tRNA. And it's actually tRNA which reads the messenger RNA. But the process begins when the ribosome binds to the messenger RNA. And then the ribosome moves down the messenger RNA in groups of 3 nucleotides that we call codons. And then the tRNA reads the messenger RNA in a group of 3 nucleotides, which is the codon. So the messenger RNA is translated into codons, and you need 3 nucleotides to code for 1 amino acid. Any question about any of that? Surprisingly, translation does not begin at the start of the messenger RNA. when the ribosome binds to the messenger RNA and then moves down the messenger RNA, protein translation does not begin at the start. Protein translation does not begin until the ribosome reaches the start codon, which is AUG. So once the ribosome binds to the messenger RNA and then moves down it, it doesn't do anything until the ribosome reaches the start codon, which is AUG. And you should probably know the start codon because there's only one. The start codon serves two functions. It tells the ribosome, begin protein translation here, and then it also codes for the amino acid methionine. So every protein, when it is first translated, will always start with the amino acid methionine because that is what AUG codes for. If the protein does not want to start with the amino acid methionine, it will remove it in probably the Golgi apparatus, where the protein is modified and packaged for delivering. We'll talk about that if we haven't already. So the messenger RNA, nothing is done until the ribosome reaches the start codon, which is AUG. And remember, besides being the start codon, AUG also codes for the amino acid methionine. And then the ribosome will read the codons as it moves down the messenger RNA in groups of 3, which we call codons. And it will translate those codons into amino acids. And that will continue until the ribosome reaches one of the stop codons. There's 3 different stop codons, so you don't need to memorize them. But one is UAA, for example. And if the ribosome ever reaches UAA, that will code for the stopping of protein translation. Any question about any of that? And this icon is showing you that translation occurs after the information in DNA is transcribed into messenger RNA. And then translation is the changing the information in messenger RNA to the information in proteins. Any question about any of that? All right, let's talk a little bit about the genetic code. Each messenger RNA has a codon which codes for one amino acid. Humans have 20 different amino acids, and most organisms have 20 or near that number of amino acids, but there are slightly over 60 possible codons. So how does the system work? Well, there's more than one codon for most of the amino acids. Let me blow this up so I can see it further. There's only 2 amino acids which have one codon. Of course, there's methionine, which the only codon that codes for it is AUG. And then the other is tryptophan. You don't need to know that, which the only codon that codes for tryptophan is UGG. All the other amino acids have 2 codons that code for them, or 4, like phenylalanine has 2 codons that code for that amino acid, or 4 codons that code for it, like serine has 4 codons that code for it, or some of them have 6 codons that code for that amino acid, like leucine has 6 codons the code for this one amino acid. So that means there are more than one codon for many of the amino acids. And this is known as degeneracy of the genetic code, where more than one codon can code for the same amino acid. Any question about any of that? All right, let's go through protein translation. I might have a video on that. That might be the video we just watched, or started to watch, and I stopped it.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
All right, any questions about that? I'll close that one. So let's look at protein translation in a little more detail. Protein translation happens when the messenger RNA comes together with the ribosome, and you don't really need to know that the ribosome is in subunits and it forms over the messenger RNA. And then when tRNA comes in and reads the messenger RNA. So all of these components are needed to begin protein translation. The ribosome does nothing at the start of the messenger RNA except read down it. And the ribosome doesn't do anything until it reaches the start codon, or AUG. Once it reaches the start codon, it begins protein translation here. And what happens is tRNA comes in and binds to AUG. What happens is the tRNA actually hydrogen bonds to the codon of messenger RNA. And so the anticodon, which is in the tRNA, will be UAC. You don't need to know that, but it hydrogen bonds to AUG. And then this first site of the ribosome makes a channel for the first tRNA to come in. And the first tRNA will always bring in the amino acid methionine, and methionine is always paired with the tRNA that has the anticodon UAC, which binds to the start codon, hydrogen bonds. So tRNA brings in the correct amino acid, and it's tRNA which is actually reading the messenger RNA. Sometimes you'll hear people talking about the ribosome reading the messenger RNA. The ribosome is allowing the reading to come, but the reading is actually done by tRNA. What the ribosome is doing is putting everything together, binding to the messenger RNA, moving down it by groups of codons, and then making this channel, which will allow the tRNA to come in. And the channel stabilizes the binding of the tRNA to the start codon, meaning that the tRNA is binding to the ribosome right here and right there. Probably hydrogen binding too. Any question about any of that? All right, the ribosome has more than one channel. There's actually 3 channels in it, but the third isn't used. The second channel allows another tRNA to come in, and this second tRNA has to be a tRNA that has the anticodon, which will bind to the second codon of the messenger RNA. And what happens is this tRNA brings in the correct amino acid that's encoded by the second codon. Any question about any of that? All right. The ribosome then removes the amino acid from the first tRNA and then takes that amino acid and binds it to the amino acid on the second tRNA. If you recall, I mentioned when I was talking about ribozymes that this chemical reaction is actually happening by the rRNA of the ribosome. So it's a ribozyme that is removing this amino acid, and then it's also a ribozyme that is attaching that amino acid to this amino acid to begin growing the polypeptide chain, which will become, when it's all finished, the finalized protein. So we can call this the start of the growing protein. And that's the only ribozyme we'll ever talk about in this class. All right. Once that methionine is taken off of that tRNA can then fall off the ribosome and the messenger RNA and then come out. And that will allow the ribosome to move down one codon. So this position will then move to that, and then this position will move to the third ribosome, which is right here. You'll note that what was once in the second position of the ribosome now moves to the first channel of the ribosome. That happens when the ribosome moves down a codon. That will allow go back here, that will allow the next codon to be read and the tRNA to bring in the correct amino acid, which happens here. And then once again, the ribosome removes the amino acid attached to this tRNA and then combines it with the amino acid over here on the newest tRNA that's been brought on. And so now we have a growing protein that has 3 amino acids. When that happens, this tRNA is now free and it'll fall out. And then the ribosome will move down. Let me go back to make sure I got that right. CCA. I don't even see CCA anymore. There it is there. The point is that once again, the ribosome will move down when this amino acid is removed from this tRNA and this tRNA falls out. And that will make the ribosome move down another codon. And then another tRNA will come in the second position of the ribosome, bring in the correct amino acid. And the ribosome will move down the messenger RNA in codons like this to bring in the new amino acid. That will continue until the ribosome moves down to a stop codon. The stop codon tells the ribosome to stop protein translation here. That will cause the ribosome to fall apart. That tRNA will come out. That will release the growing polypeptide chain, which will be the growing protein, and that will be the final protein, and it'll be released. So the messenger RNA will be released from the ribosome, the protein will be released, and then the tRNA will come out as well. That will allow this messenger RNA to bind to another ribosome and begin the process of protein translation all over again. Any question about any of that? All right, if not, I'm going to stop the lesson here and we will begin the lab. Give me one extra minute so that I can get a few things done before we begin the lab. Any questions?
No, thank you.
All right, I'll see you in the lab at 6:31.
Okay, bye.
Okay.